Okay, well, welcome everybody. Thank you for joining us in another ReefTalk. Just a quick reminder, these are brought to you by Malama Kai Foundation. And my name is Peter Heine. Most of you know me. And make sure you get on the mailing list that we have these upcoming ReefTalks. Try to offer these once a month, many times a year. So Malama Kai actually is thoughtful stewardship of our coastal waters. Malama Pono Oke Kai. And that's what Malama Kai has been doing for a long time in the community. Established in 1991 to enhance marine and coastal protection in Hawaii through education, outreach, support of community-based resource management, and current and future generations. Malama Kai Foundation is one of the longest-standing not-for-profits around the island, and I'm very proud to represent them with ReefTalks and other programs. Dave's moorings were the first buoys installed in 1990, and now over 200 exist statewide. Works with Division of Boating and Ocean Recreation and boat operators, and helps and facilitates, monitors the Dave's mooring program, which is one of the easiest ways we can help protect the coral reefs. These don't drop anchors, and pretty much you'll save coral. Simple. And then we also work with the Ridge to Reef Stewardship Program. I work with others on this and take middle schoolers from Malpa to Makai in stewardship field trips. Get their hands dirty, they plant native species and remove invasives, and that's all grant funded. We are not-for-profit. Malama Kai is a not-for-profit, so donations are always appreciated, and we're always seeking funding. As well as the ReefTalks, which are monthly, usually last Thursday, often of the month, or second to last Thursday. Resource for the West Hawaii community. And there's a lot of past projects that Malama Kai Foundation has been involved in. The ReefTalks were started over 20 years ago and funded by donations. We are hosted in this amazing facility. So grateful to be here by Arizona State University and Terraformation. And if you'd like to donate or follow us on social media, those are the QR codes, as well as we have information in the back. And so with that, I'm going to go ahead and present tonight's speaker, Mikayla Barnwell with the Nature Conservancy of Hawaii. Thank you.
All right, my name is Mikayla. I work for the Nature Conservancy. I am their coral restoration specialist. And first of all, thank you everyone for coming out tonight and hearing about our project. I want to share a little bit about kind of how we got here, what we've been up to. We have a place-based pilot coral restoration project that we've been working on for several months now. So excited to share updates on what we've got going on. So first, before we get into our project, I just wanted to go over a little bit of background focusing on coral specifically and restoration. Are there any folks in here that are familiar with coral biology or have volunteered doing restoration before, besides the people that I work with? Okay, perfect. So coral is a colonial organism. So the color that you see when you go snorkeling or pictures and scuba diving, that bright color is its tissue. It's covered with thousands of polyps, individual animals. So that's a close-up drawing of tissue is what is the live part of the coral. And so you can see huge mounds of it, but it's still only that thin tissue layer that's alive. That thin tissue layer, the color comes from a symbiotic algae that lives in its tissue. And so not only does it give it its color, but also provides the coral with most of its food and energy. So super important to its livelihood. And so through that, they can produce a calcium carbonate skeleton. And so that's what's underneath the tissue layer. That's what gives it its big 3D structure, which is super important for creating habitat on the reef. It's a food source, also important for coastal communities and protecting from storm surge and waves as that 3D structure kind of diminishes some of the wave energy. So it's super important for us. One of the things that I wanted to highlight before we talk about restoration is coral's reproductive capabilities. That's why coral restoration is so successful and we're able to do what we can. They reproduce two ways, one of which is sexual reproduction. So they release gametes into the water column. I don't know if anyone has been snorkeling in the last couple of days, but there's a species similar to this one, brown lobe coral, that has been spawning over the last few days. So there's been a lot of coral gametes on the surface of the water. If you've seen a big slick or anything, it's really exciting to see that happen. But they'll release eggs and sperm into the water column, and those will fertilize and settle and become tiny little baby corals. But corals can also asexually reproduce. So if one were to break off, or if you have a branching coral and a few branches break off, that one branch will continue to grow and become a genetic clone of the piece that it came from. So similar with boulder species, if a piece of it breaks off, the tissue will continue to grow, and now you'll have two genetic identical ones. And so that's how we make our restoration work. So getting into what is coral restoration, there's a lot of different ways to do it, a lot of different places that do it. But for what we do, it's when we're intervening with different processes, whether that's active or directly getting into the water, or more passive, we're kind of setting up management plans, but we're helping to restore damaged or degraded ecosystems and try to return some of that ecological function. And so these are all pictures, and you might see some of these again, and I'm definitely going to talk more about them, but of the restoration that we've been working on for the last 8 months or so, this is the methods that we've been using in the field. But like I said, this happens everywhere globally. Besides Hawaii Island, places in Oahu and Maui are doing coral restoration. The pictures all the way on the left side are actually from Dar's Coral Nursery in Hawaii. So the top picture is growing in their nursery. They use more of a kind of a cement pyramid structure that, like in the bottom. There's also places that do larval coral restoration, so playing to that sexual reproductive phase. They'll go out and either collect coral spawn or spawn them in the lab and settle them on different substrates that they will then outplant on the reef. So I don't know if you can see it, but there's a tiny little baby coral right there. And so they have these kind of little stars that they've put everywhere. In other places, like in Indonesia, they have what they call reef stars. So for more branching species, this is the rebar dome that they can go and put out over big rubble fields where there's not a lot of structure to recreate that reef habitat. And then similar in French Polynesia, they also have a type of coral nursery. And in Oahu also, Kuleana Corals is an organization. We work with them sometimes as well, but they're doing outplanting. And then in the Caribbean, they also have some more traditional, or what we would think of as traditional restoration structures with lines and PVC trees that they hang branching corals that grow in the water column. And so, you know, while this is done everywhere and we're not totally reinventing the wheel, restoration in West Hawaii specifically is relatively novel. And so we're working with other organizations here that are doing restoration to kind of figure out what works best for the corals that we have. So for restoration specifically in West Hawaii, one of the reasons that we started doing restoration was because of some damage from coral bleaching events in the last decade. So this is an example from some DAR surveys in Kealakekua Bay in 2011 on the left. So if you can see all the way on the left, there's like it looks like a basketball size, it's cauliflower coral. That same one is kind of over here. But with coral bleaching, the water temperature gets too hot, and that symbiotic algae that lives in the tissue starts to produce a toxin, and the coral will actually expel the algae. And so it'll look bright white. So it almost kind of looks like there's snow underwater. But because they're not able to get most of their energy and food from that algae, it often will lead to mortality. And so with the dead coral, for a little while, there'll still be structure, but there's nothing contributing to keeping that structure there, remaining strong. And so it will start to degrade, erosion. And so this is pictures from 6 years later after a bleaching event in 2015, where most of that structure has gone away. The coral population is significantly smaller. And this is just one example of where it happened in West Hawaii. And so it took a significant percentage throughout West Hawaii, and that's kind of one of the big driving reasons organizations got together to decide. And so also for why we're doing it here in West Hawaii, we work with the communities that we're doing restoration in. So we're building capacity, and we don't necessarily want to be just out there doing it and then they happen to find out about it. We work with them to do this work. And so we can kind of reconnect the people in the place, work with the community members. These are all photos from events that we have done where the community has come out and made a significant contribution to the work that we wouldn't really be able to do without their help. So that's been a huge factor in this project. This project is also super important culturally. It provides an opportunity for the community members to kind of reinforce cultural practices and stories in the Hawaiian creation chant in the Kumalipo. It refers to the coral as kind of the original ancestor for living things. And so through this project, we've been able to work with community member, kind of reconnect them to place, connect us to place, and kind of revitalize some of the cultural practices that maybe they haven't been able to do in a while. What was really special is these photos here. Before we started any in-water work, we got together with the people of the communities that we're working with to hold an opening ceremony. So we had mele and protocol and really honored more than just the ecological significance of this work. The cultural significance is just as important. So once we all kind of decided that this is work that needs to be done, we started a huge planning process to figure out what restoration needed to be done, where specifically. And like I said, it was place-based and community-driven. So we worked with the people of the place that we were doing restoration for. So we have two sites, which I'll show you in a bit. But up north in Ka'upalehu, we worked with the Ka'upalehu Marine Life Advisory Committee. And then down south in South Kona, we worked in Kealakekua. So we worked with Kapu Kapu Ohana. And we had volunteers from each of those places get together with TNC and also other partner organizations, DAR, NOAA, and we met probably like 3 or 4 times for each place to really sit down and talk about where should we do restoration in each of these places, why is it important to you, what species should we work with, what are some long-term goals, what methods should we do. So we had a series of planning workshops with the community to figure out how we should design this project. And then we were able to kind of move forward with an initial plan. But project was learning about the restoration methods that are already being done all over the world and how we might need to adapt them to what we do here. So members of my team and myself took trips to Florida to the Mote Marine Lab to learn about what restoration methods they were doing. The Nature Conservancy in the U.S. Virgin Islands in St. Croix has had a long-standing coral restoration program. That's actually where I got my start a few years before I worked here. So I got to go back and work with them and see what methods they were using and be able to bring that back to my team. We went to restoration-based conferences, and also members of our team went to CRF, or Coral Restoration Foundation in Florida. That's been also a long-standing restoration organization, and learned about their methods, some of which we've brought directly back to what we use. In this center picture, you can see kind of almost like a credit card with a mound on it. That's coral growing on a little plastic card. We've started using that for some of the corals that we grow out for some species, and we're finding that it works fairly well. So learning a lot was a huge part of the planning process as well. So now to get more into specifically kind of what we ended up deciding, what we went with for our pilot design and methods. And it's relatively small scale for this pilot project. Like I said, we're learning what works for these different species in these different places so that eventually when more people start doing restoration or we want to expand, we can move forward with the most successful method. So our 2 locations, like I mentioned before, Ka'upalehu, we have Kahuwai Bay, which is right in front of Kona Village, and then Kealakekua Bay, just down south. And so in each of these locations, we have a similar setup for our restoration project. We do want to be able to monitor and keep track and test the methods that we're using. So we have a series of plots set up that we're doing restoration in. In each location, there's 8 plots. Half of them we're doing, we're outplanting in, and then half of them are control. We want to be able to see in the end if the corals that we're working with or intervening with are either growing faster or more successful than corals that have had no intervention. So we're tracking the control as well over time. And we also have a nursery table in each of these locations. And I'll talk a little bit more about that in a bit. So when talking with the planning committee and the community members to kind of figure out what species we should work with, we kind of decided among 2 target species that we were really going to focus on. So Parietes lobata or Passalopora meandrina or cauliflower coral. Those are kind of 2 main species. Parietes lobata is really structurally complex and is a little bit bigger and more mounding, contributing to that 3D structure. Passalopora meandrina really got wiped out by the bleaching events in 2015 and 2019. And so we are working to try to bring back some of that coral tissue, some of that abundance, to put more of it back out onto the reef. And that was also an important factor when working with the communities. But in addition to those target species, working with additional species and increasing coral diversity was also important for the community. So we kind of have a series or a set of other species that opportunistically, as we come across them, we can work with them. And we've worked with a few, I think we've worked with at least 1 or 2 colonies of each of these additional species. We want to start figuring out, of the methods that we're trying out for these additional species, which one works better for those as well. So in addition to abundance, we're also looking at diversity. So in each of these places, we have, we're doing the same methods in each one. And so these are kind of our main methods that we're working with here. We have whole colony reattachment. So when we go out looking for corals, when we collect them, we collect what we call corals of opportunity. So rather than ones that are attached to the reef, we're looking for ones that are rolling around in the sand that likely wouldn't survive anyway. So we'll rescue those ones and use them for any of these methods that we're working on. We also do an underwater propagation. So with the cauliflower coral, it's a bit more sensitive than the mounding lobata. So when we collect it underwater, we'll directly fragment it on site underwater. We don't bring it ashore. And then we'll directly outplant those fragments to our restoration plots. But we also do an onshore propagation. So with the lobata, one of the methods that we're working with is fragmenting it into smaller pieces to kind of create more, put more biomass out, and then direct outplant it, or we have a nursery table and we'll put some of it on the nursery table for a grow out to be able to compare in the end if one of these methods was more successful for their growth. And so just for a quick example, this is one of our restoration plots in Kealakekua. So we do monitoring before, during, and at the end of any of our implementations for these methods so that we can track over time. And so this is before we have outplanted anything, but all of our by 10 meter area will outplant within each of these methods. And then just to show you kind of what we're hoping for to put back in the plot, these are all of our outplant types, and I'll talk in a little bit more detail about some of them. But the top 2 are related to fragmenting our lobata into smaller pieces. So the ones that we directly outplant, we're looking to put in each plot, we're going to put 6 out there. of total fragmented, what we're calling clusters or array, we'll have 12. And so the other half will come from ones that grew on the nursery table. So we'll have 12 of these cluster arrays made up of fragments of lobata in each plot. And then with our large colony rescue or reattaching corals, we'll have 12 of these for Porites lobata to start with in each plot. And then there will also be 12 fragments of the Meandrina. So while this is not as colorful as it would look like under what we actually put out there, this is kind of our baseline numbers that we're going for. And once we kind of get through our pilot push, then we can start looking at more higher numbers and additional species to add to this as well. So to get kind of more into the nitty-gritty of the methods that we're actually working with, the first one, reattaching whole colonies or large colony rescue. So this is when we're going out looking for our corals of opportunity. For this particular method, we're looking at Porites lobata. And so we're looking for a little bit larger size colonies, because when we put it back out on the reef, it's going to contribute to the structural complexity a little bit faster than if it was a smaller colony or if we're putting out fragments. So for this one, we're looking to reattach those large colonies, and we actually use a cement mixture. So in the middle picture, you can see it almost looks like the diver's holding a frosting bag, but it's a reusable piping bag that we have a cement mixture in, and we go and put a little piece out on the reef, put our coral in it, and make sure it's nice and stable. And then we can put more on the edges if we need to. But we'll use cement that we mix right on the boat into these bags and attach the corals this way. And the cement cures in about 24 hours or so. So most of our reattached colonies are fairly stable. And so this is an example of kind of day one of reattaching a large colony versus several months later when we've gone to check on it. So if you can see on the left here, I circled some kind of damaged spots that from maybe rolling around in the sand. Dead patch of tissue damage. There's a little bit of scarring. But if you look on the right side, you can see that both of those areas and maybe any other marks have healed over, and the tissue is looking a little bit brighter. And so it's really exciting to be able to go back and see that that it's healing and it's stable, and that's what we're looking for with these larger colonies. And so across both of our restoration sites in Kahuvai and Kealakekua, we've reattached 96 lobata colonies. So we have 48 in each location to go along with the diagram that I showed you. We have a few other colonies of other species we've started to work with. We have a few Porites evermani that we've also reattached. But with our main push for reattachment in lobata, we've done a total of 96, which is exciting. So for Passalacra meandrina, like I said, that outplanting process and the fragmentation process happens underwater. It's a bit more sensitive of a species, so we don't want to bring it ashore to fragment like we would our lobata. So when we're collecting them, we can also collect off of a man-made structure. So these corals on the left here are all growing on a mooring line. So we're going to relocate them to our restoration plots. So we always get pictures of what they look like beforehand. And then to fragment them underwater, we use a hammer and chisels. So the middle picture on the top here is exactly what he's doing there. So we just kind of look for the crevices and we tap it a few times to break it into a few smaller pieces that are more suitable for outplanting. And we use these kind of modified milk crates down here where we can put all of the fragments of the different parents that we're working with to bring them to our restoration site. And then we'll outplant them using 2-part marine epoxy. We use this for our meandrina outplants as well as our smaller lobata outplants. And then there's 12 of these in each of our plots as well. And then similarly, we've also worked with 96 fragments. I think we have collected like almost 32 actual colonies. And then because we're fragmenting it to break it into more pieces and put out more corals, we ended up putting out 96 fragments. So in addition to the underwater direct outplanting with the Meandrina or the large colony stabilization, we also have an onshore propagation method. And so this is kind of a really big push and is one of our bigger methods. We use this for Porites lobata and similarly other mounding corals. We fragmented a Porites evermana. Divers will go out and do a collection as normal. They'll collect corals of opportunity of the boulder species, and then they'll actually bring them on shore. We have a flow-through system that we've designed to be able to hold these corals on land for a few hours while we're fragmenting them. So we've got a holding cooler that they'll all go in when they get on land that has fresh seawater running through it, and then it'll go into a physical version of this where corals can get hammered and chiseled to be more manageable sizes for the saws that we run them through. And then they'll go into more of a data collection phase. So just to go back for a second, in the picture on the left, the instrument that he's working with is a diamond bandsaw. So we use that to fragment all of our corals into smaller pieces for outplanting. And then the picture, if you can see at the bottom, is an example of kind of where we would break it into smaller pieces for the saw. We'll also collect data on it initially before anything happens to it and record how big it was when we got it, if there's any health conditions, give it a name also so that it can be tracked throughout the process. And these events are very community-based. We worked with many school groups. When we worked up north in Kahuvai, we had a bunch of school groups that came through, and also in Kealakekua is a really big community turnout. And so with events like this in this type of project, bringing out the community to help with this work in their place is really rewarding. And we couldn't really have done this work without the community help, but also while we're able to teach them about coral propagation, we learn more about the history of the place from our community members. So it's a very rewarding process. So for our onshore propagation, once we fragment the corals into small pieces, they'll go kind of two different places. So we want to be able to see and to test if directly outplanting coral fragments or letting them grow out on our nursery table contributes to the success, their overall success. So they'll get prepped to go. We have a modified tackle box here on the left with coral fragments in it, if you can see at the bottom. The divers will take this tray down to our restoration plots with them and then directly outplant right on the reef using that same 2-part epoxy. Or on the top here, they're getting glued onto these PVC trays with some tiles on top. And these trays are made to slide right on our nursery table that you see on the bottom here. And so we always make sure to have tissue for both of these places so that we are able to directly compare. And so regardless of kind of where these corals go, they're going to get outplanted in a similar fashion. Just depends on the number of fragments in the design. So when they get outplanted onto the reef, we're going for this cluster array shape. Corals typically grow 1 to 2 centimeters a year, which is really slow. But with the fragmentation method, when we're cutting the coral, it actually activates accelerated tissue growth mechanism. And so the coral is trying to heal itself. And so by cutting the coral on each side, we're kind of kick-starting that tissue growth. And so then it will grow out from all sides and hopefully fuse together to become one colony that can, we're aiming for about 12 to 13 centimeters. So here's a little graphic of, in the end, we want it to all fuse together to become one colony. And we started outplanting at a size of 12 centimeters so that in a year or so, it'll be about 12 or 13 centimeters in diameter. And that's an important goal size that we're looking for. 12 centimeters is kind of where it starts to really contribute to kind of the ecosystem, and also could be a good spawning size. So we're able to kind of put out more corals of reproductive size at once. And so we haven't gotten a full colony fusion yet, but we have had some fusion in our fragments that we've outplanted so far. So we outplanted in Kahuvai back in February and March. And so this is an example from those outplants. So if you see at the very top, you can see there's the brown tissue on top, but then the white side is that exposed skeleton from underneath the tissue. If you move to the right for day 33, you can see that you can't really see some of the skeleton anymore, but there's a bright yellow tissue. That's new growth that has just started to kind of grow over the sides, which is what we're looking to see. And then if you go to the bottom left, you can look kind of at the bottom and see that some of those fragments, the tissue has actually started to grow together already. So you see less of that bright yellow, and it looks more kind of a uniform color. And then the last time that we've gathered a picture, monitored 138 days after it was outplanted, you can see kind of along the bottom curve, most of those fragments have started to fuse together, as well as along the top. So we're anticipating, year for these to fuse together, but this is really exciting to see just after outplanting these in February or March. So it's a good sign for what's to come. And this is highlighting their asexual reproductive process. And so this works because all of those fragments have the same genetic background and allows them to grow together. So if they're not getting directly outplanted right after they're fragmented, they go out on our nursery table for kind of an extended growth period. So it's a little bit of a safer environment, maybe more water flow, and it'll take them a little bit longer to grow out on the table. What we're looking for is those tiles that they're on here. We want to see the tissue kind of grow over the sides of the tile before we outplant them back onto the reef. And then we'll outplant them in a similar array. There won't be as many fragments because they're already a little bigger to start, but they will get outplanted in that 12 centimeter array, and we'll wait for hopefully a year for the tissue to continue to grow over the tile and fuse all together. And so what's been really exciting is across both locations that we've worked in, we fragmented a total of 1,508 coral fragments between the two locations. So in Kahuvai, we had 3 fragmenting days, we got 623. And then in Kealakekua, we had 4 fragmenting days and got 885 fragments. So a portion of these will go to grow out on the nursery table, and then a portion of them will get directly outplanted as arrays right on the reef. And so we started, all of these fragments came from a total of 48 parent colonies that we've collected throughout all of our fragmenting days. And then with the array design, outplanting those smaller pieces, it'll be over 100 arrays that get outplanted between the two sites. So you can really kind of see that increase in the number of corals we're putting out. And then for monitoring, so we do a structure from motion monitoring process. So we take a camera and we swim over our restoration plot in a grid, like a lawnmower pattern, and we're rapidly taking photos of our plot with all the outplants. And then the computer program can stitch all these together to create an orthomosaic, or what you see on the side here. And so we can use this to continually monitor and repeat this process at different time points and look for the same corals at each time point and measure their growth over time. To this orthomosaic on the computer program and actually find our outplants that we've put. And so this is an example here. I'm not sure if you can see it, but In the top right here, we have an example of a meandrina fragment that we outplanted. There's another one down here. And then kind of in the bottom center, there's an array that was outplanted. So once we kind of do the initial SFM with all the outplants, we can name them on the computer. And then as we continue to go out and do more monitoring, we can match up from the newest one and compare growth sizes or success, if they're alive or dead. And it's a really helpful process to be able to visualize this well while we're on land. And so as we've been kind of going through this process and trying out all these different methods, trying to figure out what has worked, we finished our main implementation push for our restoration project, and we're going to be looking at trying to answer these questions as we start to analyze data. And so we want to know if there's a difference in survival between our nursery grown fragments or our directly outplanted fragments of Parietes lobata. So we'll be able to compare those two since we have parent tissue, the same tissue from the parents in both of these places. And then we also want to know if there's a difference in success between the fragmented corals, so those fragmented arrays that we put out, or just directly stabilizing or reattaching a large colony. And then also thinking about restoration in the future as we move forward. What is the cost benefit ratio between nursery grown fragments, directly outplanted fragments, and reattached colonies of Parietes lobata? So as we're continuing to expand and do more restoration, it's also a cost benefit. We also want to think about that as we're doing more methods or continuing to do the same on which one was more successful for the corals, and also what makes the most sense for our organization to move forward and see if there's a difference in any of those. And so as we start to be able to answer these questions as we pick up our monitoring and analyze our data, we also want to start brainstorming what do we do next? What's next? What species? What methods are we going to work with based on the results that we've gotten from this pilot project? And what other ways can we engage our community in this work? Thank you. Yes.
How scalable is this? It looks like it's very intensive and work out and do this work. You've got a couple of small plots. Big coastline.
Yeah.
Is it scalable, do you think?
That's definitely one of the questions that we're working on trying to answer as we do think about expanding. Our team is fairly small, and we rely on volunteer divers and community members for some of our events. But I think as we move forward, thinking about which of our methods is most successful can kind of help us guide in that direction. If one of our methods is more sustainable for us to do and it's helpful for the coral, then that might contribute to the scaling up factor as well. But also what's nice is that our organization is doing restoration, but we work alongside DAR and ASU. We're doing restoration in the same locations. They're doing slightly different methods, but we're all kind of able to try out methods at the same time as a collective group and contribute to more areas than we could do alone.
You mentioned the Parietes lobata spawning at the beginning of your talk. Is there a formula for that? Is there a number of days after some period of life cycle?
So I believe the coral that spawned this month was Parietes evermani, which looks very similar. It's in the same genus, but it's in any coral spawning, it's dictated by the moon phase and number of days after sunset. So spawning typically happens in the summertime when the water is a little bit warmer. But you want to look at the full moon, and I'm not super familiar with the spawning cycles of corals in Hawaii. I know in the Caribbean, they have like a huge chart and table where it's like 2 hours after sunset, this many days after the full moon, and it can get pretty specific. But from what I know, we're still doing a lot to learn about when corals spawn here. So I think the people down in Ka'alu'u were able to see this coral spawn last month and this month, which is really helpful data. Yeah, yesterday as well. So it spawned yesterday, and then I want to say a similar time of the month in June. And so using that, you can kind of predict moving forward. So next year may not be the same exact day, but you might have to go out maybe one day before the full moon and a day after, and just kind of hope you can see it. So it's definitely a trial and error process to figuring that out.
So I thought it was really great and also interesting that when you have your outplanted tiles, you have like a tag. You can them by computer. What kind of tags do you put on?
So we have kind of tried a few different ways. So if you can see here in this picture, we have a yellow cattle tag next to some of our outplants. And so the way we first tried it was temporarily putting a tag next to coral so that it has a name. And once we monitored it, we went back out and removed all of those tags, so we weren't leaving them there. But we actually found it was more beneficial to use these painted fishing weights. So we don't leave these on the reef for any longer than we're underwater that one day. But between the people that are outplanting and collecting data, this fishing weight essentially moves with the coral all day. So we have on land, this parent colony is fishing weight number 3, and that gets written all the way until it gets outplanted. And so then we'll grab a picture of it like this. And also we do our structure from motion monitoring on the same day. So in that orthomosaic, you can zoom in and find the fishing weight. And we're able to remove all the fishing weights as soon as we do the survey that one day. And once we have that orthomosaic, we can look back at our data sheet. And once we zoom in and find that this coral has weight 3 next to it, we know from what we wrote down that it's coral number whatever. And then we can just put that on the computer.
For some reason I thought, as you said, that's put them up by computer, but you have to actually visually, it's not right there.
Yeah, we have to go in and manually input the names on the first time that we do it. And once we kind of, that we take a lot of data throughout, but especially at the beginning when we want to make sure that we're recording our corals properly, so that it gets a name and that name stays with it throughout the fragmenting process. So we know that all these fragments came from the same parent. And when we go to put it on the computer, it'll be accurate.
Do you have a method of determining where you're going to reconnect the corals? I mean, you're not planting corals on top of other corals.
Right. So to get to that point, when we're in the water, setting up our plots, we're looking for viable outplanting space, which is bare substrate. So depending on the location, there's more or less of it. When we were in Kahuvai, there's a bit more coral cover up north. So once we set up our plot, we're kind of looking for viable outplanting space in all of our plots, even though some of them will be controlled, because we'll decide that afterwards. But then in Kealakekua, there's a lot less substrate to begin with. There's a lot of rubble there. So picking where we put our plots maybe is a little bit more choosy. But before we even get in the water, we've looked at maps where they've figured out what the substrate is so that we have a general idea of, based on this map, they're saying this area is more of this kind of substrate, and then we can go with our site visits and actually look at it ourselves to make sure that this is where we want to outplant corals. Mm-hmm.
I understand that the bleaching event of last summer in Florida, most to nearly all of the corals that had been restored, outplanted over the last 20 years were wiped out. Is there any reason for optimism? Are there exceptions to that that give you cause to have a hope in the long-term outlook?
So one of the things that we're looking into as we move forward with this project and start expanding is kind of trying to identify which corals may be able to survive higher water temperatures. So other places have done thermal tolerant tests on corals, and you can see naturally in a bleaching event if some corals will bleach completely, some maybe pale a little bit, some maybe won't bleach. And so we can see that some are more resilient to these higher water temperatures. And so with our restoration, what we want to know is, are any of our corals more thermally resilient to bleaching? And so we're working to kind of set up those tests where we can put a few fragments in an experiment to see what temperature they're able to withstand. And then results from that maybe will kind of guide us on which species or colonies that we can use for restoration moving forward that may be able to withstand the higher water temperatures and not bleach as much.
As far as practical results over this last year's global bleaching event, have there been spots of optimism where outlanded corals did survive?
There's definitely have been some that have survived. I'm not super familiar with the Caribbean or in Florida's reefs at the moment, so I don't know specifically where, but I do know in the Nature Conservancy also works on an island atoll, Palmyra Atoll, and they went through a really big bleaching event as well. And most of their corals had bleached, but from what we're starting to see now, there's been a significant amount of recovery. So while it's happening, it's unfortunately one of those that you just kind of have to wait and see once the bleaching event is over what survives. But there is a lot more that survived than we were expecting. So, Grace?
So you mentioned that one of your goals of restoration is to reach larger size colonies. Are you guys planning on, and so larger size colonies, in the long term monitoring how the amount of sexual reproduction in that area is changing, how many babies are being produced?
From the corals that we've outplanted?
Right. So like are you seeing more sexual reproduction in Kealakekua after restoration?
I think like looking at reproduction of what we've put out is something that we've thought about for like future expansions of the project, and we definitely would like to do, or at least I would like to do, some larval work in our project. And so I think in the future, once the corals that we've outplanted have been there for a little while, starting to look at their reproductive capabilities next to what has already been out there, I think is super important, because that's kind of like the full cycle of restoration, is putting out a coral and it grows big enough and healthy enough that it can then reproduce on its own, is what we're going for. So definitely something to look into for the future. Mm-hmm.
So actually last night at dinner, Guy asked me, well, why don't they just take corals from warm water places like the Red Sea or Indonesia, where these corals are really tolerant of higher temperature water, and transplant them here or in the Caribbean?
I know that there have been a few studies. I think the one I can think of was in Australia, where they did do kind of a transplant experiment and moved corals from different water temperatures. For here specifically, since working directly with the community members, we're working only in the places that we're restoring. So only taking corals from Kahubai Bay for restoration in Kahubai, and only taking corals from Kealakekua Bay for that restoration, rather than bringing in corals from other places.
So is there a scientific reason?
It may have worked in some other places, but I know a lot of people are kind of on the fence about it, to bringing in invasive species. Not 100% sure like how they'll grow in that new environment. So they could either completely take over and kind of a high competitor with what's already there. So the coral species that are native there or natural don't have as much space or aren't able to grow as well compared to the new species. So it's always a risk bringing in something from somewhere else and hoping it'll grow the way you want, because you can't always be sure of that. Yeah. Mm-hmm.
What year did you start doing planting?
So we started field work in the beginning of last November, so November of 2023. And for our pilot phase, we've kind of just gotten to the end of our main implementation push at the. So we've only been in the field since November doing this, but we're going to be continuing to monitor, do maintenance on our nursery while we're monitoring our outplants and seeing how they grow. Mm-hmm.
So it's not quite a year.
Correct. Yeah.
Okay, well, let's get to those reporting part of the project for you personally, and then follow up as community members how that's going to support you.
Thank you. I think one of the most rewarding moments, and I kind of have this on every dive, is we're out there and, you know, actually putting the coral back on the reef and just like picking it up out of the rubble and putting it in a place where it's has a better chance of survival. And also seeing the tissue fusion already. Like I get excited every time I see these pictures because it's growth and it's growing back together, and we have a whole bunch of these on the reef, and to see them have to fuse together in multiple places is really, really rewarding. And we're always looking for volunteers to come out and help. It's harder to get in the water with us, but a lot of times where we need help is on land, either with our like big fragmenting days or even on construction days where we have a whole bunch of trays to make for our nursery table or we're making different things. Our team is pretty small, so we always look for extra hands to put together stuff like that. Any other questions?
So do you live here on island?
Mm-hmm. Yeah, so I actually moved here in 2021 for my master's degree at Hilo. And their tropical conservation biology program had a professional internship track, and that's how I got started with the Nature Conservancy. So I was brought on as an intern and kind of helping with the planning and development of this project. And then the following summer, I joined the team as a full-time employee, and so I have been working on this project ever since. So it's been really exciting to kind of see this project go from paperwork to fieldwork. So
do you live still in Hilo, or you?
No, last summer I moved over to Kona, be closer to all the field work. Any other questions? Thank you. I'm